Hey guys, so this is your midterm review topic 2 of protein synthesis. So protein synthesis is literally how the body makes proteins. So synthesis means make, so we are making proteins. The central dogma, which is like the central belief of how proteins are made, are DNA to mRNA to protein. There are 2 steps within this. So the first step is transcription, which is DNA that produces mRNA, and then the second step is called translation. And that's when you start with mRNA and then you end with protein. So let's go over this a little bit. I'm going to show you in 2 different ways. So if we're starting with DNA, okay, so this is just basic structure of DNA. All right, to make proteins, I literally take a section of DNA. A section of DNA is just called a gene. So every gene has a specific sequence of nucleotides. And it is within that specific sequence of nucleotides that determines the mRNA that is transcribed, and therefore the proteins that are made. Okay, so remember, each of these little circles represents one amino acid. One codon codes for one amino acid. If I have a bunch of amino acids strung together, that's what creates a protein. So I start with DNA. I just take a chunk or a little section of DNA, which is called a gene. The specific sequence of nucleotides determine the RNA that's transcribed, which therefore determines the type of proteins that's made. So basically, if anything within this sequence of nucleotides changes, that means that my protein is also going to change. And remember, my nucleotide is just the building block of DNA. Okay. So if we look at it from a cell, so let's say I have a cell. I'm going to move this down. Cell. Okay. So the outer rim is a cell membrane. Inside, okay, I am starting with my DNA. So this is DNA. And then my DNA, because, okay, so, sorry, this inner circle is the nucleus, because that's where DNA is kept. My first part of protein synthesis is transcription, when I'm converting DNA to RNA. So I'm just going to draw a little picture. Okay, so DNA I know is a double helix, RNA is a single helix. I'm converting DNA to RNA, or I am transcribing. So this is first transcription. All right, after transcription, my now mRNA, M is for messenger, so this little messenger RNA is going to travel outside into my cytoplasm. And it is going to find a nice little ribosome. This is not to scale, by the way. It is going to find a nice little ribosome in the cytoplasm and basically deliver this message that DNA gave it about what kind of protein to make. So this is mRNA. All right, now mRNA has these little letters. Okay, I'm just going to do 3 letters. These represent the bases on it. Okay, so every 3 letters is 1 codon. Okay, my tRNA, this is going to be my tRNA, basically goes and finds the little amino acid that matches the codon here, and it drops it off. So my codon here was AUG. And then it's going to go and find this little amino acid here. And then basically just add it to the growing chain of amino acids. They just keep dropping things off. This is my amino acid sequence or my protein. Okay. After we've done that, this whole process here is translation. That is the part where I've started with mRNA and I end with protein. Okay, now I want you to go to another page. Okay. If we were to take an example, all right, let's say we had a question that says, what is the protein made from the DNA sequence ATGCTA. Okay. So this is giving me DNA. I know that this is DNA because it has thymine and RNA has no thymine. My first step of making a protein is transcription. Transcription is when I start with DNA and I end with mRNA. I'm starting with DNA and now I need to go to mRNA. Whenever I am transcribing, I'm going to use the base pairs adenine to uracil and guanine to cytosine. So adenine pairs with uracil, thymine, adenine, guanine, cytosine, cytosine, guanine, thymine, adenine, and adenine to uracil. Okay. Now that I've made my mRNA strand, I need to divide my mRNA strand into codons. So remember that 3 bases, which is what each of these letters represent, 3 bases equals 1 codon, which equals 1 amino acid. Now I get to use this fun little chart that all of you love. Okay, so I'm only going to use what is on the mRNA. So my first codon is UAC. To read this chart, I'm going to find my base on the very left. This is U. That means that my answer needs to be somewhere in this row. My second base is A. So that means that now my answer needs to be in this box. And then my last letter in my codon is C, which now means that there's going to be in this box in this letter, or in this box in this row. So my first codon, or not my first codon, my first amino acid is TYR, which stands for tyrosine. Let me find my second one. All right, my first letter is G, so I know that my answer needs to be in this row. My second one is A, so I know that it needs to be in this box. And then my third letter is U, so I know that it needs to be up here, so that is ASP. So all these little letters simply represent like abbreviations of amino acids. When I have amino acids put together, that makes a protein. So this is going to be my amino acid sequence, which is my protein. And this is what my answer would be. So once you finish this, show me your notes and then you can get started on the practice.